It's wonderful to be here. Thank you for the introduction. It's as David mentioned, I did my PhD here, and it's just great to be back. And it's a nice opportunity to also reflect on how much the faculty members in this department had influenced me, including Dave Bougie, who's here. I really appreciate all the, particularly now as a faculty member, all the great training I had as a graduate student. I know there's a lot of graduate students here, so you'll appreciate your faculty probably even more after you leave the university. But so really fortunate to feel I've had such an influence from Paul Nogdy and Mike Carroll, Stan Berger, Dave Bougie, and I really appreciate that. So thank you. So today I'd like to talk to you about some of the work in my research group at the University of Pittsburgh. It's largely focused on soft tissue biomechanics with an idea of trying to look into the structure and understand how it affects the function of the materials and also how these things change in disease and also in healing. So a big area of my group is working on cerebral brain arteries as well as brain aneurysms. In addition, we've done some work on in vivo tissue engineering of blood vessels, where we've worked with Yang Wang of Cornell University to look at what happens when we put a scaffold inside of an animal and the body comes in and builds a vessel as that degrades away. And I'm also going to be talking a little bit today about the bladder, which is some recent research that my group has become involved with. So we're trying to do this in a multimodal way where we're using bioimaging coupled with experimental systems as well as modeling tools and with some of my collaborators, computational tools to try to address these issues. There's a lot of work right now in collagen-based biomolecules. So these are the main building blocks of these soft tissues are collagen and elastin. And there's a lot of work recognizing that we really need to understand this structure in order to be able to treat diseases properly. So it's fascinating that even going back as far as 1847, that Wertheim recognized that these soft tissues kind of had a qualitatively similar behavior. What you see here is actually Wertheim's data plotted on one plot where the strain scale is different for these different tissues as well as bone. And the idea is that Wertheim recognized that this J-shaped curve, which we have come to know and appreciate if we work in arterial mechanics, is something common to all these different soft tissues and uncommon to bone and inorganic materials. So at that time, of course, he didn't have Instron machines. He's literally putting weights on strips of soft tissue, but it was a beautiful piece of work that's still recognized as quite impactful. And so a lot of our work has been trying to understand some of these curves and how they change with disease and how this kind of structure that leads to this can affect the failure of tissues. So just to give a little bit of background, the main structural components from a mechanical point of view of these tissues is collagen and elastin. So the collagen is a hierarchical structure. We have the molecules, of course, into fibers, into fibrils. Fibrils and fibers. For this talk, we'll mainly be focusing on the fiber level. But when we look at damage of these tissues, we really do need to go down to these micro scales. So what you see here is an SEM of the fibrils and some work from our group with the multiphoton microscopy of the collagen fibers. And then this is actually a cerebral aneurysm that we see here looking at the structure of that. The elastin also has this hierarchical structure where you can have fibrils making up a fiber, as in this SEM picture on the left, but they can also form in lamellae and even have lamellae which are attached by elastin fibers. So in terms of understanding the function of this, a crucial factor is that the collagen is orders of magnitude stiffer than the elastin. And it's in fact orders of magnitude stiffer than the tissue itself, in particular that toe region that I was mentioning for this J-shaped curve. So that brings up the question of how can these be combined to create something with these J-shaped response. So as early as 1954, it was known by Burton, or recognized or conjectured, in fact, that this combination of elastin and collagen could produce that if the collagen was recruited in a delayed manner. So if we start loading this tissue, the collagen doesn't actually sort of unfoil and become recruited until a finite level of stretch. And as a result, then you can get this kind of J-shaped curve. So this was the conjecture. Burton and Roach did some work, very nice work with using enzymes to digest out individual types of extracellular matrix components. So for example, this is the intact vessel. We can see what happens when you remove the elastin. It becomes stiffer, just as expected, because collagen is the main component now bearing load. Or we can also dissolve out the collagen and get this much softer response. So Lanir came up with a constitutive model for this in 1979 and 1983. And you might wonder why I'm still talking about a constitutive model from this long period ago. But it turns out he was way ahead of his time. So he was proposing a constitutive model which we could not actually measure the components for to feed the material properties into this. And in fact, if you go through the literature, even now, and since 2019, I think there's been like 130 citations of this paper And you can see that it's now becoming in more and more prominence and more recognized for its value. But I'm going to talk about the constitutive model in a moment. I just want to give you a sort of, let's say, qualitative idea of what's going on here. The idea is if we take a kind of imaginary strip of some length, unload it, and we load it, the fiber waviness, and there's some fibers that are more wavy than others, will begin to straighten out. And so there'll be a moment when the fibers are considered load bearing. It might not be perfectly straight, but there might be, some reason that they're now load bearing in that let's say, level of tortuosity. So this is something that Lanir cleverly incorporated into his model. It turns out we found that it's playing a major role in the response of the materials is this gradual recruitment process. In addition, as you'll see in a moment, he also included variation of angles in his work. So if we go back to a more conventional presentation of this idea of multiple reference configurations, we can see here is a figure from some of our multiphoton work of an unloaded tissue. You can see this tortuosity. It's very evident. And as you stretch out this material, so associated with the fibers in that reference configuration, you can have a direction at each point that's giving you a sense of orientation of this tortuous fiber. And then as the fiber material stretched, that sort of imaginary material element will be in fact elongated. And at some point, it'll be elongated sufficiently that this fiber will be able to be load bearing if you add any more stretch to it. So this is another reference configuration, which is very important for describing the material then, because it's only beyond this level that the fibers actually load bearing and engage in the process of contributing to load carrying for the material. So this is something then that was built into the constitutive model that was proposed by Lanir, in slightly different presentation here. Initially, he'd had it with a summation where you could have a countable number of fibers, or you can take the limit where you have an integral. But basically, you have a strain energy function per unit volume, the reference configuration, that has a dependence on the current stretch of that filament. But in fact, that has another integral defining it, which accounts for this sort of recruitment of this tortuous fiber. So there's a distribution function for the angles because their fibers can be at many different angles. There's also a distribution function for this recruitment stretch. And so these are the things in principle that would need to be modeled to be able to use this constitutive equation effectively. But this is something that we did not have the technology to really do at the time. And that's one of the reasons it's only recently that this is becoming more and more used, as well as the fact that with more computational facilities, power, we can begin to look at complex loading conditions with a constitutive model like this. So just to go back on that, prior work trying to, let's say, provide data for this kind of constitutive model, there was some work done very nice work by Michael Sachs using small angle light scattering to try to get information about the tissue. And he's done a lot of work on, for example, heart valves. But we have another of different alternatives right now in terms of technology. One, which is still a little bit unideal, and I'll talk about that in a minute, is to just basically take 2D sections where we can use stains and things to see many different aspects of the wall. The thing is, it's not naturally in 3D, but if you do serial sectioning, you can in principle build back up a three-dimensional tissue structure. And there's been work, for example, by Sylvia Salfeld to try to do that using various machine learning algorithms. The other thing we can use is confocal microscopy, which is literally getting sections of information in the plane. And that can be very useful for trying to understand the through plane response of the material. However, unfortunately, this is the only thing we can see there without staining or fixation is the elastin layers. With the advent of being able to use multiphoton microscopy, though, we can also look at collagen without staining or fixation. So this is actually a human brain aneurysm. You can see these interwoven fibers that exist in this remodeled tissue. So there's a number of reasons why we're fortunate we have access to multiphoton microscopy rather than just confocal, because the longer wavelengths, we can have greater penetration. We can also use conventional optics. We have less photo damage. But the main thing really is that we can look at both collagen and elastin without fixation. So that opened up the possibility, which we were very fortunate to be able to do, which is to build mechanical devices to put under this microscope, because now we could see how the collagen fibers are being recruited to load bearing during this loading process. And so this is work that we are very excited about. So just as a minor illustration to see what I'm talking about, in the same sample, which is very important in biological tissues, which have so much variability, we could look at different stretch levels. So we can see that, for example, here, let me see if I can use this, for example. We can see what happens when we have no stretch. We can see this clear tortuosity in these medial fibers, and we can also see them being stretched. So we'll be able to quantify this and to be able to come up with constitutive models for this. So the first thing we were able to see is that even just qualitatively looking at this data sets, we could look at how these different levels of tortuosity corresponded to this loading curve. And we can see that just qualitatively that every single fiber we can see in the sample, for example, is tortuous. And as we continue to higher and higher stretch levels, we can see that they're being recruited. So this is just a qualitative look at this, which verifies this idea that there's this delayed and gradual recruitment of collagen. We have to reach a finite stretch before we're going to see collagen recruited. So the first thing we wanted to do is to populate or understand more about the angle distribution in these arteries. And so this here is just we can see right here, this position here, we can see this sort of en face view of the medial collagen fibers, which we can trace and start to quantify how they're located. But we can also see if we take them, because this is a stack of images, all with fibers traced in them, we can now make an artificial cut there and see how the fibers are lined in the orthogonal plane. And we can see that they're really lined, this has been flattened, in the circumferential direction. So there's not fibers going out of the plane. So we can really start to think about making a constitutive model that has 2D layers of these collagen fibers, rather than having at every point some kind of three-dimensional contribution of fibers, which is important. So we're basically seeing we have a 2D display distribution of these. So we, just for the possibility of getting the best possible data, we did this angle evaluation after the fibers were really relatively straightened out, and then mapped them back to the unloaded configuration to get this distribution inside the reference configuration. So with this, we were able to get this basically this PDF of this distribution of fiber angles. The next thing was to try to understand how we could get this recruitment stretch. And so basically, in order to finish this constitutive model populating with data, we need to be able to understand this this PDF of this distribution of gradual recruitment for these fibers. And so essentially, we're trying to understand the fraction of fibers that are recruited in some infinitesimal change in the recruitment stretch. So by measuring the tortuosity in the fibers, we were able to get a sense of how the various distributions of tortuosity vary at different stretches. And so you can see here tortuosity levels as a function of stretch of this tissue. And we can see that as we get higher and higher stretches, the tortuosity is heading more and more toward 1. And we have a critical cutoff for this recruitment of these fibers so that they're going to be load bearing. And that is something that I'll talk about a little bit later how we chose that number. But this is the critical tortuosity level. So then once we have that information, we're basically populating a CDF. So because we're looking at the cumulative distribution. So at any given stretch, we're going to see how many fibers have already been recruited. What we really want for this constitutive model is the PDF, and that's a simple mathematical calculation to convert to a PDF. So basically, we used a beta distribution function because it has a finite range. And we believe that the fibers start at a finite stretch, and then they also complete recruitment. So this then, just as an overview of this, we were able to get the fiber distribution of the angles to get this tortuosity distribution, which gave us this PDF of the recruitment distribution, and also get then mechanical response. So a critical point is that we only have one fitted parameter. We're not inputting the stiffness of these fibers. We're fitting that to the overall bulk response of this material. And so this worked very well for these samples. We had 8 samples. We had a very good fit for them, which was really exciting because basically we were able to predict this part of the curve. All we're doing is fitting that slope. So everything else was obtained from imaging data, with the exception of the elastin contribution, which was fit to the low level loads. So this brings up an important point. We've got a lot of constitutive equations which are more phenomenological or don't account for this finite recruitment stretch. And so they're a little bit less physical, right? Because they're going to have a long exponential response of the collagen fiber. And in addition, we know that collagen fibers are going to have some kind of damage to them after about 20% strain, which means that we're essentially going to be, if we want to look at failure of something, we're going to have incorrect information about the failure process because we're going to start the fiber straining at the unloaded configuration of the body, and so they'll reach much higher strain values. So this is something we've taken advantage of in various constitutive models to look at failure processes. So just to give you a sense, sort of new directions we're heading in, we can also take a finite element approach. Work is done by my collaborator, Spanid Maiti, and graduate student. But basically, we can embed these fibers based on the experimental data that we just discussed for tortuosity and fiber angle directly into finite elements, and then look at, come up with a constitutive equation. There's a simple linear case here, but we can come up with a constitutive equation that enables us to look at failure of that material by having this multi-layered composite that models what we see in the artery. So we can have a fiber network based on measurements we've made directly with multiphoton microscopy, as well as information about the areal density and the thickness of these various wall layers. So we can really try to begin to understand directly what's going on with these tissues. So just to give you an idea of what I mean by this is, here's the data we have in blue. That's the mechanical testing data of stress versus stretch for these arteries. We can use the multiphoton data, et cetera, to build this computational fiber model, and then we can stretch it and look for how it fits the data. So once again, we have a constant associated with the collagen stiffness and the elastin stiffness, but other than that, the shape of this curve is being determined by measured quantities. And so this is important to be able to go beyond this elastic regime and look at the failure process, because we know from our prior testing on elastic tissues that we can have all sorts of failure characteristics. I mean, this is just a qualitative picture, but we can have abrupt failure, or we can have these various kinds of gradual failure. And so this is something that we're beginning to look at the role of this gradual recruitment in generating such a gradual response. This gradual response is particularly important for biological systems because you don't want something failing abruptly, right? Because then it's catastrophic. An aneurysm failing abruptly in the brain is going to bleed out. The person's not going to make it to the hospital. But if you have this kind of failure and more gradual, the biological system, just like if we cut our hand or finger, can go in and start a healing process going on. So I'd like to talk to you now a little bit about some of our work on cerebral aneurysms. So shift into how we're using these kind of approaches to study cerebral aneurysms. This is part of a large multi-institutional work that we're doing to try to look directly at the aneurysm tissue, do bioimaging and mechanical testing on it, and come up with constitutive equations for it. We've been fortunate to have funding where we can collect samples from 4 different sites and get over 164 samples now. What I want to focus on today is a little bit about the time progression of these changes in the orientation and tortuosity of biological tissues. So one of the things about cerebral aneurysms, which we can see here, it's a brain aneurysm, it's an outcropping of part of the vessel wall, is we can see that its shape can change relatively quickly. In about 18 days, we have this dramatic change in shape. And so the body, as I was just mentioning, has to be able to respond to these changing geometry and try to reinforce itself. Shown next to that is an example of a fusiform aneurysm with a bleb on the top, just to give you a sense that we can have saccular aneurysms, we can have fusiform aneurysms, we can have these outcroppings. We see many different geometric presentations of these aneurysms. So what I want to talk to you a little bit about today is actually just being able to study the changes in this structure over time. So the idea that if we have an animal model, we don't just get one sample from a person, we're able to get samples at many different time points and get a sense of how things evolve in time. And this is one of the things we want to do in terms of stabilization of the aneurysms, is to understand more about that. So this work I'm going to talk about right now is part of research that we have with the Mayo Clinic with David Kalmus, who developed this animal model of an aneurysm, where basically they're putting in elastase into this wall to dissolve away that component. They get a saccular shape then, and then they can test devices on it, look at different processes, and in our case, look at how things are changing in time. So you can see this is the basic idea of how it's made. In the figure next to it, we can see what it looks like in terms of the lumen of that after it's formed. So in terms of the mechanics and what the cells are experiencing in the wall and how they're responding, basically what we see is that this aneurysm here, which we can see right here, was formerly the vessel, which has an arrow pointing next to it in blue. And so in that stature, we could even use some simple Laplace law to estimate it, but we could also calculate it to get more information about the stress distribution based on the local geometry. But we can get a sense of the relationship between the axial loading along the length of the vessel versus their circumferential. So in a typical case like this, there's a lot more circumferential loading. We can see it's down around 0.2, 0.3 in terms of this ratio of the axial load to the circumferential. But once the aneurysm is formed, this changes markedly, and the axial load becomes more like the circumferential load. So in terms of the wall, what it has to do, it has this change in load in the wall. The cells are experiencing that, and they're going to try to remodel in order to be able to have a condition that's better able to bear these biaxial loads. So just to give you an idea what the original state is at, let's say, time t equals 0, we have a wall which has multiple layers, has a media, it has an adventitia. In the adventitia, you can see there's these red collagen fibers. The media has these green elastin lamellae. And behind that, which we can't see, are also collagen fibers. In this state, before we've changed anything, we can see that also the fibers are largely lined and they're more distributed, but they're more, let's say, aligned slightly more in the circumferential direction. And then we have the media, which typical of blood vessels, the medial fibers are mostly circumferential to counteract this inflation, the stresses in the wall from inflation. And so the question is, like, what can the wall do after this change? And this kind of question of growth remodeling response of the wall, this is a very, very common question in biomechanics. We see this in many different settings, how people respond to hypertension, how they respond, and I'll talk about a little bit later, to bladder outlet obstruction. Many different biological systems are looking at how the wall is responding to load. And so in this case, what we found is what we chose to do is we couldn't do biaxial tests on this because of the size, but we could do uniaxial tests. So we wanted to see how the capacity of this wall to bear that an increased axial load changed in time as a result of remodeling. And basically what we found is that we can see the control artery, that it was very soft, right? So under this, roughly 1 megapascal, for example, it's up to a stretch of 2, versus the aneurysm, which 8 weeks later, it's remodeled and it's much stiffer than this control. So something changed in the wall that changed the mechanical properties dramatically. And so what we wanted to do is to go in and understand what the wall was capable of doing and how it was able to make a difference in terms of this capacity for bearing load. So once again, we're going to use multiphoton microscopy. We're going to look at the different layers through the thickness to see how things are remodeling. So what we found, first of all, is that if we take the control artery, the media fibers are not able to bear this axial load. I mean, very little. So they start off largely circumferential. And you can see this here by these heat maps that basically most of the fibers are in the 0 or pi direction, meaning they're circumferential. And then as we increase the stretch, there's a little bit of engagement, but not very much. And you can see that also, that even up to a stretch of 2, there's very little engagement of the media. The adventitia of having a wider distribution of fibers is more able to contribute. So it's probably contributing most of the capacity for this wall to bear this axial load. As you can see, the fibers go from more circumferential, and eventually you have this more axial loaded fibers as we increase the stretch. So this is what the normal control artery could do. And so now we're asking, what possibly can change for this aneurysm wall? How can it restructure itself? How can it rebuild this wall to have a better capacity, like we see here, to be able to bear this load? And so one of the things we found, which was very striking, and I don't think has been seen in other scenarios, is the fact that a new layer was generated. Most of the growth remodeling theories that you'll see out there are really designed to modify existing layers. Okay, maybe the fiber is reoriented to be in a direction over time that's more suitable for the load bearing. What we see here is a kind of fast response to address this need to bear load in that direction. It's kind of like if something dramatic happened to your house, you're not going to start remodeling it. You might just put up plywood quickly to stabilize things and then remodel it, right? So that's what we think is going on here. By 2 weeks, 3 out of 5 of them had already had this new layer, because that's the first time point that we imaged at. By 4 weeks, 5 out of 5, and by 8 weeks, 6 out of 6. So this was common in all these remodeled blood vessels. So this was very striking. And we also then said, well, okay, they were laid down, but can they actually reorient and bear load? And the answer was yes. If we put them in a uniaxial system and look to see how they were load bearing, we could see that in fact they could reorient and bear load and contribute to load bearing. The other thing we saw then is what was more conventionally noted is that over longer time periods, the existing fibers restructure themselves to be able to contribute to this modified load. And so in this particular case, we see the media at 8 and 12 weeks, these later time points. The media went from being largely circumferential all the way through the wall to now having a region near the blood or near the lumen where it was in fact orthogonal to that it was in the axial direction. So it's now has this kind of ply where the layer that's at the top there is now able to bear the axial load, and the other fibers are more or less maintaining their original direction. And this is something that we evaluated. Well, can they reorient and realign to the loading direction? The answer is yes. So in summary, this part of the study addressed how the vascular wall can remodel in response to pushing the load from largely circumferential to more equally biaxial. We saw a 2-phase response. It was located between the adventitia and the media, which is interesting that was a location that has a lot of implications about cells that exist around there to do this kind of remodeling. And we have multidirectional fibers. And then a phase 2 where the media fibers are actually remodeling. So the data obtained here, we developed a constitutive equation for, I'm not showing it here for sake of time, but basically we could use it to populate, drive the development of a constitutive model for this single time point. And we're also now working on developing growth remodeling theories that can capture this kind of remodeling over time. And I think it's also very important that we can then use this animal model to try to understand how to enhance this remodeling in other situations and try to explore a little bit more what is a deterrent to this remodeling. Please.
So it's not, I understand correctly, that's a elastase model. So you remove most of the elasticity.
Yes.
It's not just a response to increased load, but also a response to the absence of elastic.
Right. So, yeah, so the elastin is basically removed. There's sort of an immediate shock. And then by 2 weeks, it's deteriorating rapidly, probably not contributing to load by that time point. So that's the loss of that. A question, a loss of elastin, so at that point, basically the wall has nothing to really contribute to axial load except for this reorganization, realignment of the adventitial collagen fibers, because the elastin layers appear as lamellae.
Yeah.
So they're naturally suited to handling load in any direction.
Right.
So once the elastase is put in, those layers are removed, and now you're largely left with medial collagen fibers and adventitial collagen fibers. So that's what we're looking at is now that we've changed the load and we've removed this potential load-bearing component in the axial direction, what is the body going to do? Why we're interested in this elastase model is because, in fact, I'm glad you brought this up, aneurysms, brain aneurysms, with almost no exception, few exception, don't have elastin in them. And so this is also a good model for looking at something like that. Okay, thank you. Okay, so the last topic I want to go to is one that we've begun to work on relatively recently, which is the bladder. So I'm just kind of giving you, examples where this kind of focus on structure and function are very important and have implications for clinical events. So maybe some of you know about bladder outlet obstruction. Basically, the prostate gland can enlarge and sort of diminish or increase the resistance in urethra. And as a result, the bladder now has to generate a larger pressure inside to drive the urine out of the bladder. And that's kind of like, that's possible because one of the ways the body can remodel is to build up muscle content to be able to generate this larger pressure inside the bladder, just like we can, build up muscles in our arms. We can also, the vessel walls can do that as well, including this bladder. So that's really good. But eventually over time, that effective remodeling starts to degenerate where the wall starts having other problems, probably having to do with ischemia. We're investigating this now. But this is a problem that's quite prevalent. And as a result, we've endeavored in going into this research in a kind of larger scale manner. We've already published one paper on this whole remodeling process, taking data from a rat model over time and developing a growth remodeling constitutive equation to describe that data. But that's really at the early point where there's not a lot of negative effects of having this increased wall thickness, increased bladder size, et cetera. And so what we're doing in this research is trying to combine together with our colleague at Pitt, Dr. Yoshimura, who has an animal model for bladder outlet obstruction, with some of our bioimaging work and analysis and modeling, with a colleague also at Sheffield who's an expert in modeling, growth remodeling in aneurysms and also in arteries. So we were fortunate to get funding in that direction. And what I'd like to talk about today is that one aspect of that work, which is to look at the compliance of the bladder, because we're trying to understand both the filling and the voiding process. So in the filling process, what the bladder is capable of holding, this is an example of a rat bladder outside of the body where we're looking at this increase in lumen volume. So essentially, the inside of this bladder can increase more than 20 times with a relatively small change in pressure. But this has to also be done somehow in a way that the cells which are lining the bladder are not overstretched. So just like arteries have cells lining them, our skin has cells lining them. If you start to stretch something too much, you're going to put a lot of load on the junctures between the cells and on the cells themselves. So there's somehow the bladder has to be constructed in a manner where that's not happening. And obviously these cells that line it are playing sort of a control over what goes in and out of the wall just like the endothelium does for our arteries. So just to give you a little bit of background about the role of the extracellular matrix in the bladder is different than the arteries in the sense it kind of has an extra layer. It has, in addition to this lining layer, which would be the endothelium in the artery, have a urothelium, which you can see is sort of folded there. We also have this lamina propria, which is mainly collagenous fibers and cells. And then we have this detrusor layer. So in the artery, the detrusor layer has smooth muscle cells. In the artery, those two are in one layer. And this is important. The reason I'm mentioning that is because these need to be coordinated. So somehow the body, because they're both going to be load bearing and you want them to both contribute, somehow they have to be coordinated in a way that they can both be part of this filling process. Otherwise, you'll have kind of a poorly designed bladder wall, and I'll come back to that. And then just like the artery, there's an adventitia that's kind of playing the role of preventing overstretch. So what we wanted to do was to directly analyze the wall, like we did for the artery and the aneurysm wall, and try to understand how these different parts of the wall structure are altered by aging and how this thing functions. So this is the doctoral student who did a good majority of the work I'm going to talk about today. So prior to this work, there was a little bit of, let's say, conjectures out there about what was going on. It was felt that probably the lamina propria was the only thing expanding, and the detrusor layer was somehow very loose and just waiting to, help out if there is overloading of the bladder, which then there's also an adventitia, so they'd both be playing that role. So in order to be able to understand this, we set up we designed a biaxial system that could go under the multiphoton before we had a uniaxial system, so that we could look at the fibers under the multiphoton, but we could also measure the strain directly. So we have a mirror underneath this biaxial system where we can get the strain, and then we can make obtain mechanical data for the stress versus the strain. So this is something we ran an experiment with 9 male rats to look at what's going on. So we're comparing aged and young rats and trying to understand the compliance in each of them, but also how the compliance varies between these two different types of bladder. So in this case, just like we talked about with Wertheim's early experiments, we see this J-shaped curve. And we can see that we have this toe region, we also have a transition regime, and then we have this high stress regime, which basically prevents the bladder from overstretching. What we did is to try to understand this, we quantified this by defining a bound on the transition regime of a low strain level and a high strain level, where we're able to characterize this shape. And using a k-means clustering approach, we were able to then distinguish, take our data and separate it into two groups, a type 1 group and a type 2 group. So the type 1 group, you can see, has a longer, softer toe region, and the type 2 group is more quickly recruited to load bearing. So one factor, so we're just how does the bladder have some of this capacity for high compliance? One aspect of this is these rugae, the so-called rugae, which are basically folds in the surface. So it looks, to us, like sort of a buckled surface. And that seems very good in some ways because it's possibly one mechanism for not overloading the cells that line that surface. So our first question was, can we measure how this, undulated surface is being recruited and flattened out during this study? So we used the multiphoton microscope to get these layered studies to find, and we could then see that surface in these layers in the stack, and we could figure out how it basically diminished these peaks and valleys to identify when it was flattened out. And so you can see here, this is one of the images we have is really a stack of images, and we could then have a heat map of basically the topography of this. And so what we found is that these rugae, in all cases, because we're also running mechanical experiments, did not flatten out until this transition regime. So they're basically, that's a crucial part of it. It was very consistent among both aged and young rats. And so we think that they're, providing this mechanism for preventing overstretching of the bladder. The other thing we then were concerned about is how does this vary and differ between these two groups of bladders that we're looking at? So if we just consider this range here, this range of recruitment, or this, let's say, transition regime, and where these rugae were flattened, we can look at that same data set for all the rat cases. And what we could see is that the one case, which was had more longer toe region, which is up there, type 1, where this region was longer, that's visible in this, let's say, graph. But we can also see where these rugae were flattened there. There was always in this transition regime. And then we have type 2. These are generally older animals, as you can see here. There was one adult, and I guess we didn't see in this room, probably, young adults, some people are going to look older. So this is really a phenotype we're looking at, but it could include young animals in addition to the older animals. And similarly, some aged animals could have a younger phenotype. And so that's how we categorize these in this study, is really by phenotype, not by age. And so that was one mechanism, this rug-eye flattening. The other thing we wanted to do is understand the collagen recruitment process and how that came into play. And so here we basically, as before, we looked at the collagen organization at different levels of stretch. And what we could see is that basically, as we get to this high stretch region, we see most of the fibers have been recruited. And in the detrusor muscle layer, which is now a little bit deeper in the wall, it has muscles in addition to collagen. We can see them, they're highly tortuous fibers, and they're also being recruited. So this then brought up the idea of, can we somehow compare when, we want to see when they're recruited in this whole process. So we basically started tracing the tortuosity, try to quantify this, and then plot, once again, this kind of CDF of the percentage of recruited fibers at different levels of strain. And so what we found is that there was never any recruitment in this toe region, that as we might expect, the collagen fibers had to be delayed in their recruitment in order to have this nice flexible region, whereas most of the recruitment occurred in this transition regime. So we could then basically try to start to compare the PDFs of these. So we could take this data, generate a cumulative distribution function, which is telling us the fraction which has been recruited by these different stretch levels, and then get this PDF, which is telling us what fraction are being just recruited at each different stretch level. And we could, come up with an equation for it, put it in our constitutive models, et cetera. But what we found is, which was interesting, is that in the case of this type 1, which is highly flexible, that this recruitment was highly coordinated between these two different layers. So we have the detrusor layer, we have the lamina propria layer. The detrusor layer is the one with muscles, but there are two layers that, like I mentioned, when artery just has the one media, these two layers both seem to be engaged in the normal physiologic regime. And in the case of the older rats, or this older phenotype, let's call it, what we saw is that this was no longer coordinated, but now the detrusor layer was prematurely recruited. And this could even be seen in the multiphoton images, where this is this layer here, where we see that we have a highly tortuous fibers that are not recruited and some which are recruited. And that's what's going to give us this very long regime, is that we have things recruiting very early, and then there's still things being recruited at a much higher stretch. So this helped us very much in understanding what was malfunctioning with this bladder. So in summary of this bladder, we basically have this toe region, just like other soft tissues, just like Wertheim had recognized. We find that in this toe region, what's happening largely is that collagen is not being recruited yet, but it's the rug eye are being flattened. And then in this transition regime, what we have is we have recruitment of both the detrusor and the lamina propria, but in the type 2, that it's a prolonged or sort of early recruitment, and it's prolonged over a longer strain regime. So this was the first, was very striking evidence of this extensive toe region. And we've developed, I haven't discussed them, but constitutive models for this. And, we sort of see now how we can try to model this difference in terms of the lack of coordination between the layers. And future directions, we're going to include the active response. So far, we're just doing voiding, but we want to do, filling, but we want to do voiding, and as part of this larger project, look at pharmacological treatments for bladder outlet obstruction. So I want to show you here a couple new directions we're going in with this. So far, what I talked about is the bladder where it was somewhat already filled. So we're not talking about when it's completely voided down, but something that's partially filled, and we're looking at how we can continue that latter part of the filling stage. But very recently, I was just talking with David about this. We developed techniques where we can look at even fresh bladder under multiphoton and see the folds that occur in this highly voided state. So now we're in a position where we're going to try to understand this folding process and understand basically what happens during filling as well for this complex system. So, we're looking into the collagen fibers, how they're organized around these folds to try to understand this process. Just another, sort of comment about where we're going with this. One of the things we're doing with cerebral aneurysms I didn't talk about is we're looking at trying to improve risk assessment. Why do aneurysms fail? Why are they some rupturing and others are not rupturing? And how, and we think there's multiple reasons. Some of them have inclusions like calcification. Some of them have very thin walls. Some of them just have stress points or points where it's difficult for the collagen fibers to remodel properly. So this is just giving you a picture of like a failed aneurysm. So we're doing a lot of work to try to understand this failure process. And this is a picture of an aneurysm where we can see, for example, in the dome of the aneurysm has biaxial fibers, which we might expect for something which is dome-like. There's also applications of this focus on fiber orientation in some work I'm just going to show you from one of my junior colleagues at Pitt, who's done some work with collaborators at Harvard and Irvine and trying to remember the other university where they're coming from. Zurich. And so basically what they're doing is they're building a scaffold, which is kind of an artificial heart ventricle, and they using that system to explore why these fibers are aligned in nature, so biomimicry, in this helical manner versus circumferential. And so this is an example of where they've actually seeded these little sacs that they've created, ventricles that they've created with myocytes so that they can contract down and they can compare the pumping volume, et cetera, in these different systems. So I think it's really cool, this idea of using an experimental system to explore the importance of different conformations of fibers and things. So that's where I think it's a good stopping place. Just want to thank you for your attention. See if people have any more questions.
You have time for a few questions? Yeah, thank you very much for the nice talk. I have a question on the first part.
Okay, sure.
The elasticity model of material, which is fiber structure.
Yeah.
And you probably have like a fiber network model. How was it set up and how did you choose the size of it? Model?
You mean if that computational model was built?
Right, goes like a system of interacting rods.
Right. Okay. Right. So yeah. So in that case, there's basically fibers. So if you have a finite element, you're embedding fibers in that. The fibers are generated, let's say, shot out to be able to put them in the same kind of distribution function as exists in the tissue. So you're going to have some sort of distribution of fibers in that model. And then it's assumed that there's an affine deformation so that the fibers themselves are tracking with this finite element in terms of their, let's say, kinematics. So that's currently the assumption behind that. And then, of course, the size of the element things can be considered. This was just like a small volume that we're considering this for so far.
Yeah, if it's affine, just to be careful because it's overshooting, you cannot make it. So uniform strain assumption.
Yeah, right. Yeah, so that's the assumption currently, correct. Thank you. Thank you.
On a similar topic, so you showed the strain energy functions depending on lambda f and lambda t. I guess the total deformation gradient for stretch and total so the incremental one. So all of your modeling is one-dimensional at the constitutive level, or are you, the fibers then many dimensional, two-dimensional, but essentially one-dimensional fibers, or are you thinking of this also as a two-dimensional constitutive model?
Okay. So yeah, so basically let's just take one plane because we can then have multiple planes. So in one plane, we have a distribution of fiber angles. Each of those fibers has a capacity to bear load in a single direction, which is part of the constitutive equation for that fiber. What was its orientation in the unloaded configuration? So that then, the response of that fiber when it's stretched, because of the, way that the tissue is loaded, it might rotate, but we're still overall just looking at the stretch of that fiber. That's the, let's say, driving component of the constitutive model. So in other words, if a fiber was at some 45-degree angle, we pull the tissue like this, the stretch of the tissue is in this direction, those fibers will rotate and it will be accounted for what is their actual stretch in the direction of the fiber. That stretch is what comes into the constitutive model because the fibers are considered to be, each one having at any given point one direction and one capacity of their load. In addition, they're not don't have the capacity to be compressed, so they're going to have zero resistance to compression, as assumed.
The stretching elasticity, but no flexible thickness and things.
Right. So there are some models like these helical models and things which try to bring in some of that. At this point, we don't see a driving force to do that. We do, I haven't shown it, but in the adventitia, the fibers are more helical, some of them. So that could become interesting. I think these fibers, which are more like a wet noodle going in this, it's questionable whether we really need to regard bending stiffness, but perhaps in the adventitious it maybe comes in.
Any other next?
Okay.
So a couple of questions.
Uh-huh.
You showed the recruitment of the fiber.
Mhm.
So a question that appeared to me was what would be the length of these folds? How long are the folds? And is there a distribution of how can one account that?
Right.
That was one
Mhm.
question. The other question I had was soft tissue. They always have some level of there is a biochemistry of the pore fluid.
Mhm.
There's fluid everywhere.
Mhm. Right.
How does that come into the picture, particularly when you're, say, resolving out or taking out some parts of it, for example, if you're taking out the elastin and the collagen itself,
Mhm.
how much of that is the biochemistry of the fluid, for the overall biomechanical response?
Right. Okay. Thank you. So the first question was about this tortuosity and how, for example, the amplitude and the wavelength of this, let's say, tortuosity comes into play. Correct. Okay. So basically, what we're doing is when we trace the fiber, you can kind of think of a mismatch between the arc length of this fiber and its end-to-end length, right? So you can imagine if you had two different things that you ripped and then you pull them, you'd have to stretch a certain level to get this tortuous flatten out. So what we're doing in terms of our model is we're basically assuming that there's no resistance to this flattening out. And so it doesn't matter the amplitude of this or the wavelength, it just matters the tortuosity level, which is basically the arc length of that fiber relative to the underlying direction. I showed you that vector, which is capital M in this case from the reference configuration. So that number comes into play. Now, we are kind of bringing that back in when we look at the adventitia because we're not directly measuring the CDF, we're measuring the PDF. So we don't want to overload the medius, and the adventitia is so wavy that we never see it all unfurled. So we can't calculate a CDF, we need to calculate a PDF. So we basically map that tortuosity back to the unloaded configuration in that point. And in that case, we are trying to account for a little bit of variability along this fiber. Parts of it might be helical, parts of it are more two-dimensional to account for some of that. The question about in what ways are we accounting for, let's say, fluid motion within this tissue? We are not currently accounting for that. So this is already somewhat of a, relatively, we're picking what we think is the most important component at the moment. But I would love to talk to you more about, how we can assess how important that is for the kind of study that we're doing. Right now, we're kind of assuming it's a second order effect compared to what we are including in the model. But I would love to talk to you more about it and hear your thoughts on that.
Time for one more from Ben.
I have to.
So, I was wondering to what extent you've used director theory for your modeling your fibers, which I know you learned it.
Yeah, I've been dying to use director theory. I've used it actually for arterial modeling. Yeah, and I know that actually you've looked at fibers with using director theories and have the paper not too long ago. So I was hoping to talk to you about that. But yeah, go ahead. So how would I bring director theories into that? Yes.
Okay.
Not yet.
Yeah. The other thing was, what is the mechanism whereby these systems form a new layer in order to change it? Is it similar to how the body heals its injuries in all of the other ways?
Yeah. So this is, at this point, I think we're the first to discover the right time trajectory to identify this and have the tools to look at this. So this is something relatively new, seeing this new layer form. What we're expecting is that some of the cells from the adventitia are probably coming in and laying down fibers, but we don't have, I can't say for sure because this is speculation, but it probably is a lot like other healing mechanisms where you cut your hand and fibers are laid down, cells move in there and, lay these fibers down and then, disappear again. So I think there's probably, biologists we could collaborate with to try to understand the flux of cells that come in to lay this new layer down. I think you're absolutely right. It is probably related to healing process. Yeah.
Yes. I just want to comment and like your multi-layered composite model.
Okay.
I learned from your later. My question is that why we're only interested on the tensile test
Mhm.
for the
Okay.
soft tissue.
Mhm.
What would be the difference if we have compression or torsional test?
Mhm.
Do we need that or we have to combine with the bonds to do that?
Okay, yeah.
Yeah.
So here, what we've done is we've tried to pick out, so for example, with the bladder, we know that that's a very equally biaxial type loading situation. So we designed this biaxial system to go under the microscope and test the tissue in that condition. In the other case of the arteries that were like an artificial aneurysm was made and we're analyzing that, we chose a uniaxial experiment there because we felt that the dominant change that this wall was being exposed to was an increase in axial load, and we wanted to directly probe that. The samples are relatively small, so we couldn't readily do a biaxial test the way we want. We really wanted to focus on how did its capacity to bear axial load change. Now, in terms of, I think you mentioned torsion and compression. The situations we're looking at so far, we don't think that compression is as important as what we're studying at the moment, even though I would love to talk to you more and you know, perhaps there are settings that we would be important to study it in, but so far we have not studied that. And maybe one of them is the fluid transport within the wall. In terms of torsion, again, it seems like maybe an interesting loading condition, but it's not one that we think is directly physiological for these systems that we're looking at the moment, even though it could be used to probe other aspects of what are these fibers capable of. So appreciate the question about that. Thank you.